Hello everybody, I'm Kateřina Fialova, and in a few upcoming minutes I would like to tell you something about removal of cyanobacteria amino acid from water by activated carbon. And now let me please briefly introduce myself. I am a PhD student at the Charles University in Prague in the Czech Republic, where I study the environmental sciences and protection. I also work in the Institute of Hydrodynamics of Czech Academical Sciences, where I focus on my research investigating the process of adsorption during drinking water treatment. This is my first conference where I would like to present you the latest information from this field, and in the future I would like to continue in this direction even in more details. So that was enough about me, and let's start with the main topic of the presentation, and that's the adsorption during drinking water treatment process. In this picture you can see the conventional drinking water treatment process. Based on the water quality are included the appropriate steps. Not all the steps are always necessary, but nowadays our planet is facing a problem of eutrophication, which is the process of enriching the water with nutrients such as nitrogen or phosphorus. And as a result, the cyanobacteria and green algae are developing really enormously, not only in recreational areas, but also in drinking water supplies. This is a real photo of one of reservoir in the Czech Republic, and it's a huge problem. Why exactly? Because part of this algal organic matter originating from cyanobacteria and green algae is not able to be removed by conventional drinking process based on coagulation. And moreover, with chlorine creates these substances, the disinfection byproducts. That's why we need to add adsorption onto activated carbon as an additional treating step even before the hygienic security, to prevent the production of these usually carcinogenic DBPs. Adsorption is a very complex process where the concentration of undesirable substance present in water, so-called adsorbate, decreases its concentration due to another solid substance, so-called adsorbent. That is the general definition. The complexity of this mechanism lies in several influencing factors, such as properties of the adsorbent, usually activated carbon, the nature of the adsorbate, and the solution conditions. The principle of the adsorption process is that the adsorbent, thanks to its unique properties, as you can see in this picture, full of pores of different size and high specific surface area, is able to catch the adsorbate, the matter we want to remove from water, through different mechanisms. Very often they are applied, for example, electrostatic interactions between oppositely charged adsorbent and the adsorbate, or via hydrophobic interactions. The other influencing factors that I already mentioned are solution conditions, and one of them I investigated in more details: the effect of the solution temperature and the adsorption of algal organic model substances. For my batch adsorption experiments, I chose two types of granular activated carbon with different chemical and physical properties. You can see them on these two pictures. I chose three different amino acids that were found to be the most abundant in the low molecular weight of algal organic matter, and also were not be able to be removed by coagulation process. Moreover, these three substances also have different chemical properties, and the effect of solution temperature, specifically 10, 18 and 25 Celsius degrees, were investigated hand in hand under different pH values, specifically at 5, 7 and 9. On these two graphs, we can see results of the temperature effect on the arginine removal by batch adsorption experiments at pH 5 on the left and at 9 on the right. The removal efficiency increased with increasing pH as well as with increasing temperature. The removal efficiency also differed a little bit by used activated carbon. Arginine is a strongly basic amino acid, and that is why it was removed better with picabiol, because this activated carbon has much lower point of zero charge. The dominant adsorption mechanism of this amino acid is electrostatic interactions, what was confirmed by the fact of strong pH dependency. In case of phenylalanine, it's something else. Phenylalanine is a hydrophobic amino acid. As we can see, the removal efficiency is far more higher than we could see in case of arginine. Moreover, there is almost no difference between the two examined pH values. On the other hand, the effect of temperature is much more pronounced in case of phenylalanine in comparison to arginine. This was probably caused by the hydrophobic nature of phenylalanine. The dominant adsorption mechanism in this case was hydrophobic interactions that are independent of the solution pH. The last examined amino acid is aspartic acid. This substance is very hydrophilic. That means it's very complicated to get these amino acids from water. As we can see on these two graphs, the removal efficiency is low. The pH 5 seems a little bit more suitable for its removal, and at this pH we also can see the effect of temperature. It's the same trend as I already mentioned. With increasing temperature also increases the removal efficiency. To sum up, the results of my research show that the solution temperature had an effect on the adsorption efficiency of arginine, phenylalanine, as well as aspartic acid. And this effect was different depending mainly on chemical nature of amino acid, the surface properties of used activated carbon, and the pH of the solution. It seems that the pH of the solution is more important parameter concerning drinking water treatment process than the temperature. It is due to that the pH value determines the protonation or deprotonation of functional groups present on both the adsorbent and adsorbate, and thus it controls the manifestation and extent of the interactions and significantly affects the adsorption efficiency. The overall greatest adsorption efficiency was achieved for phenylalanine. The effect of temperature solution was most seen in that case, followed by arginine, and finally aspartic acid. The different results were seen in effect of pH. Arginine was mostly affected by pH, followed by aspartic acid, and finally phenylalanine. Apparently, if the dominant adsorption mechanism is based on interactions controlled by the solution pH, then the effect of solution temperature on adsorption will not be very apparent. This behavior was confirmed in the case of arginine, which was adsorbed mainly due to electrostatic interactions that are strongly dependent on the solution pH. So in the case of arginine, the temperature had smaller effect on its removal. On the other hand, the effect of temperature was most noticeable during the adsorption of phenylalanine. The dominant mechanism in that case was hydrophobic interactions, which are minimally affected by the pH, so the effect of temperature was more pronounced. The main take-home message of this research is that adsorption of amino acids as model substances of algal organic matter is endothermic in nature, and in comparison to solution temperature, the major influential factor is the solution pH. In practice, this is a really positive finding for water treatment plant, because during the summer, when algae organisms are in the greatest growth and the amount of problematic matter is high, then the adsorption of these substances is enhanced with higher temperature, and we can get tasty, safety water. That's all from me. Thank you very much for your attention. I hope you enjoy this presentation, and if you have any question, please contact me. I will be happy to answer. Thank you very much. Have a nice day. Bye.